Here, we will learn about stem cells, how stem cells are different from other body cells. We will learn the function of stem cells in embryos, in adult animals and in plants, and how we might use stem cells as part of treatment programs to help people with different medical conditions. First, let's begin by looking at how stem cells are formed in humans and other mammals, beginning with fertilization, when an egg cell and sperm, also known as female and male gametes, fuse to form a single cell zygote, the beginning of a new organism. The zygote cell divides and becomes a hollow ball of cells. This is the embryo. The outer cells of this ball will become the placenta that feeds the embryo, while the inner cells of this ball are the embryonic stem cells that will differentiate to form all of the specialized cells of a body. Importantly though, even when you are an adult, some of your stem cells remain, but now as adult stem cells. Adult stem cells are different to embryonic stem cells. They are still undifferentiated cells, but they can only differentiate into specific set cell types, rather than any other cell type in the way that an embryonic stem cell can. For example, your bone marrow is a good source of adult stem cells, but those stem cells can only differentiate into blood cells and cells of the immune system. However, scientists now think there may be a tiny number of adult stem cells in most of the different tissues in your body, including your brain, muscle, and liver. This will be really important for repair, because while many of your differentiated cells can divide to replace themselves, some cells can't do this. In those cases, stem cells that can stay inside the adult body for years, they would become really important to be reactivated at times of disease or where tissues have been injured. Understanding stem cell biology is important and can help scientists and doctors in the future to treat disease and injury. Many people are unwell or even die because parts of their body stop working properly. For example, spinal injuries might cause paralysis because spinal nerves cannot repair themselves. People with type 1 diabetes need to inject themselves with insulin regularly because specialized cells in their pancreas have stopped being able to produce it. Millions of people would benefit if we could replace diseased or damaged body parts. In 1998, there was a breakthrough in understanding stem cell biology. Scientists managed to culture embryonic stem cells that were capable of forming most other types of cells. The hope is that cultured embryonic stem cells can be encouraged to grow into almost any other type of cell needed in a body. And in fact, this has already been tested in laboratory animals. For example, paralyzed rats injected with nerve cells grown from embryonic stem cells have been able to recover some movement. And since then, trials have been carried out injecting nerve cells grown from embryonic stem cells into the spinal cords of paralyzed human patients. These first human trials have been really small scale, but have shown that the procedure is likely to be safe, and it is believed that in the future it will be possible to help some people who have been paralyzed to walk again. In another success story, in 2014, doctors transplanted embryonic stem cells into the eyes of people that were going blind due to macular degeneration. This small-scale study was conducted just to actually check whether or not the technique was safe. However, all of the patients found they could see better, and so larger scale clinical trials are now taking place to develop this treatment. It is possible that in the future we might be able to grow whole new organs from embryonic stem cells to be used in transplant surgery. And many conditions from infertility to dementia could eventually be treated using stem cells. In fact, Chinese scientists have already reported using mouse embryonic stem cells to make sperm cells, and these sperm cells were able to fertilize mouse eggs. As we've mentioned in a previous lesson, stem cells from plants are much easier to isolate. Large numbers of stem cells are found at meristems, growing points in plants, and they can be used to make clones of parent plants very quickly and very economically. This gives us a way of producing large numbers of plant clones reliably and safely, producing large populations of identical plants for research purposes, for crops, and as a way to save rare plants from extinction. Producing large numbers of identical plants from the meristem's stem cells is also widely used in agriculture, generating large numbers of crop plants with selected features. For example, for disease or drought resistance, or for taste or for yield. In fact, every banana you eat is produced by a cloned plant.